All right, so phospholipids are going to be a type of lipid, and they will be basically found across the cell membrane in order to regulate what's entering the cell and what's leaving the cell. Now, a phospholipid is going to be made of two parts: the hydrophilic head and the hydrophobic tails. So the circular area is called the polar head or the hydrophilic head, while these tails here, okay, they are called the hydrophobic tails. They're hydrophobic, they basically hate the water, but the head is hydrophilic, it likes the water. So now the question is, why is there a polar head? What is the composition that makes up this head to be polar? So here we have the head part. It's going to be hydrophilic. It loves the water. And the reason is because it has a phosphate group and it has glycerol plus a choline. Knowing that this choline exists is not required. Just know that the phosphate and the glycerol combined would make this head a hydrophilic one that would like to mix with the water. But the tails are going to be hydrophobic because they have a lot of carbons and also a lot of hydrogens, and that would make them into a nonpolar tail. Now, as you can see here, this is going to be a saturated fatty acid because we only have single bonds between the carbon atoms. But here we're going to have a double bond of the cis type, which will cause bending of the fatty acid, and this bending is going to be responsible for lowering the melting point. And now a fatty acid with a cis double bond is going to be called an unsaturated fatty acid. Now, phospholipids will always be found in two layers. So the head, then the tails of the first layer, followed by the tails, and then the head of the second layer. And I imagine a lot of them, just plenty and plenty of them basically arranged right next to each other like this. And so essentially we can now say that the interior of the phospholipid is going to be a hydrophobic one. Hydrophobic interior of this cell membrane. So a cell membrane must have a hydrophobic interior, as shown here. And this hydrophobic interior is going to give a hard time to hydrophilic things to go through it. So things like ions and polar things like water or glucose will have a very hard things to go through that interior part. So they simply cannot go through it without requiring extra help in the form of a channel. And of course, because the heads are polar, you will have this blue area, which is the aqueous environment, which basically means that water is going to be found there, and you're going to be having this blue area or this aqueous environment wherever the heads are. So like you were told earlier, the phospholipids have to be arranged in a bilayer, basically. In a bilayer with the hydrophilic heads facing on the outside in contact with the aqueous solution inside the cell and also outside the cell. While on the other hand, these hydrophobic tails will be forming the interior of this bilayer. Next up we have a steroid, which is going to be again a type of a lipid. And they're going to be super nonpolar because they have a lot of carbons. A big carbon skeleton consisting of 4 rings. You must be able to identify a cholesterol once you see one. Right now this is a cholesterol, which is a type of a steroid. How would you identify it? By having 4 rings, basically. Having 4 rings tells you that it's a cholesterol. And of course, by the way, here we have 1 carbon, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27. We have 27 carbons in cholesterol with more hydrogens, okay, in the 40s basically. This will make cholesterol very nonpolar, and cholesterol is found as part of the structure of steroid hormones, okay? That means that steroid hormones will be themselves also nonpolar. Next up, we have proteins, and proteins will be involved in multiple functions. Protein is going to be the macromolecule, and the monomer of a protein is going to be called the amino acid. Amino acid is going to be the monomer of a protein. Now, some proteins are going to be very important by acting as enzymes. We call them enzymatic proteins, and they will also be called catalyst. That basically means they will speed up the chemical reactions, and they will make them faster without themselves being used in the reaction, without themselves being chemically used in the reaction. Now, you must be able to distinguish between a polypeptide and a protein, because ultimately a polypeptide is not functional, not yet at least, while a protein is functional. A protein, to be functional, must have undergone folding, but a polypeptide has not been folded yet, which is why it is not functional yet. Okay, a protein is made up of one or more polypeptide which have been folded, which have been coiled into a 3D shape. Yes, we can also say that a polypeptide is going to be a 2D shape, while a protein is going to be a 3D shape. So the monomer of a protein is called an amino acid, okay? And amino acids, please, you actually have to know how to draw one. This is the general structure of an amino acid. You will have carboxyl group on one end, to be also called the C-terminus. You must have the amino group on the other end, to be called the N-terminus. And you will have carbon in the center that's connected to an H, okay, as well as an R group. R is not an element. You will never find this in the periodic table. R is actually called a side chain, And because we have 20 different amino acids, that basically means we have to have 20 different R groups, because R groups ultimately tell you what amino acid we are talking about. Okay, so some amino acids will be hydrophilic. That means that they like the water. Some will be acidic hydrophilic. Some will be basic hydrophilic. Some will be neutral hydrophilic. And some will be simply hating the water, which we refer to as hydrophobic. Now, proteins have multiple functions, enzymes to make reactions faster. And you see right now here we have a substrate. A substrate is basically the substance that is going to be bonding with the enzyme for, maybe in this case, splitting this covalent bond into two parts. Okay. They can be used for storage, like the ones that you found in eggs. So eggs have a lot of proteins in them, which was supposed to be going for the embryo, but we eat it instead. Some are going to be acting as hormones, and of course hormones are able to be transported in the bloodstream to regulate things. For example, insulin. So insulin is going to be a big one that's going to be responsible for regulation of the blood glucose levels, to bring them down from high blood glucose levels to low blood glucose levels, or at least normal, by putting that glucose inside the cells. Some proteins will be responsible for the movement, which will most likely involve actin and myosin for the contraction and also relaxation of the muscle. Some are going to be involved in fighting disease in your body by acting as antibodies to basically catch the bad bacteria and the bad viruses to destroy them. Okay. Some are going to be involved in transportation, such as the transportation of oxygen inside your body by hemoglobin, or by the transportation of substances across your cell membrane, such as a transport protein, which can be used, for example, in this case, for transporting a hydrophilic substance from high concentration on the left to low concentration on the right of the cell membrane. And some proteins will be acting as receptors to receive a specific signal, okay, to receive a specific stimulus. And those receptors have to be bound by something called signaling molecules. Some proteins will be used as structural support, for example, the ones that are found in hair or skin. Here I show you the 20 different amino acids, which basically have 20 different R groups or side chains. And you actually have to be able to identify which one is hydrophilic and which one is hydrophobic. And so to do so, I would recommend knowing the hydrophilic ones, and the hydrophobic ones are going to be the other ones that you simply don't know, right? So if you know the hydrophilic ones, and if you see one that does not look hydrophilic, then it has to be hydrophobic. So how do you know if the amino acid is hydrophilic or not? Look at the side chain. If it's negatively charged, it's acidic hydrophilic. If it's positively charged, then it's going to be basic hydrophilic. And if it's not acidic or basic, it will be simply neutral hydrophilic. And you have to know what's inside those green boxes. We have 1, 2, 3, 4, 5, 6. No one's going to ask you to draw them. You just have to be able to identify them. Identify if they're hydrophilic or not based on knowing what's inside this green box. And that basically means that we have 6 hydrophilic neutral, and we have basically 5 hydrophilic acidic and basic. So we have 11 hydrophilic in total, which will basically leave you with 9 hydrophobic ones that are afraid of mixing with the water. Okay, whenever you want to join one amino acid to the other one, you must position them so that the carboxyl group of one amino acid faces the amino group of the other amino acid. Then they can join and lose water in the process by a dehydration reaction. Okay? And the bond that forms is called a peptide bond. You keep doing this, you get yourself a polypeptide. You do some folding, some coiling, some 3D shape, you get yourself a functional protein. So yeah. So in this case, for example, peptide bond is formed between carbon and nitrogen. Not any carbon though. The carbon has to be connected to a double bond, basically. Okay. As you can see, one end is the carboxyl, one end is the amino group. You get water in the process, you get a covalent bond, which is also called peptide bond. So the carboxyl group ending is called the C terminus. And the amino group ending is called the N terminus. Okay. So let's see. A protein structure is made of 4 different levels. 4 different levels. But most proteins only have 3 levels. Only some proteins require a 4th level. Only some. So primary, secondary, tertiary, all proteins must have those. Only some proteins must have the 4th level, which is called the quaternary level, or the quaternary structure, which is based on the connection of 2 or more polypeptide chains. A very common example is going to be a hemoglobin protein. It's one of the few proteins to have this type of structure, quaternary structure. Now, primary structure of a protein is basically very simple. It's just made of a sequence of amino acids. Nothing very important. They're only joined to each other by a bunch of covalent bonds. Just peptide bonds. And what determines the primary structure? It's going to be the DNA sequence. That's the one to determine the primary structure. The DNA sequence. And of course, you must have one end with the amino group, one end with the carboxyl group. Okay.
Mr. 1 second.
Okay, so the primary structure is going to be determined by the inherited genetic material that you get from your parents. And of course, the primary structure is going to be the one that dictates how the secondary one looks like and how the tertiary one looks like. Okay. So never underestimate the primary structure of the protein. Next step is going to be the secondary structure, which is going to be stabilized by hydrogen bonds. Between what? Between the atoms of the polypeptide backbone. The backbone. That's very important here. So if we're discussing hydrogen bonds of the backbone, right? Then we're discussing the secondary structure. We have two types. We have the alpha helix. We also have the beta pleated sheet. And if you guys recall from last lesson, a hydrogen bond will be forming between the H that's positively charged
And the negative charged.
of the O or the N
Or the F.
or the F. Exactly.
What's the backbone though?
Like the backbone, okay, 1 second. So that's the backbone here. So it's like a bunch of linked amino acids. And now as you can see, a hydrogen bond is just the dots. So between this backbone and that backbone to keep them attached to each other. Yeah. So this is the oxygen and that's the hydrogen. That's the oxygen, hydrogen, and so on. Okay. So the hydrogen bonding of the polypeptide backbone between the partially negative oxygen and the partially positive hydrogen. And the hydrogen has to be attached to a nitrogen. Okay, so the hydrogen that's on a nitrogen connecting with an oxygen. Partial negative, partial positive, a hydrogen bond. Okay. Thirdly, we have the tertiary structure. Most proteins become functional by having this type of structure. But this one will have many forces involved. So please pay attention. You see, now it's time to discuss the side chain, which basically means the R group. Now the R group determines this type of structure. It's because of the interactions of the R groups of the different amino acids. So one interaction is called a hydrophobic interaction, which reminds me of van der Waals forces because it's between nonpolar side chains. You see here, when you mention this type of structure, you will also see the word folding a lot. Folding is only correlated with this type of structure. Okay. And if it's a hydrophobic interaction, that's there to save it from the water. It wants to be out of contact with the water. Okay.
But aren't what is it? Polypeptide proteins also fold? Like they have to fold?
There's folding.
But what is that, primary or secondary?
Folding has to be tertiary.
Oh.
Yeah. If you see folding, it has to be tertiary. Now, we have multiple interactions. One of them is going to be hydrophobic, which means nonpolar R groups, and they will avoid the water, you see, by van der Waals forces. So you're going to have some region of the protein inside, away from the water that's on the outside. This is because of hydrophobic interactions. We're also going to be having hydrogen bonds between the polar side chains. We also have ionic bonds between the positive charge and the negative charge. Okay. So yeah. We also have a covalent bond called disulfide linkage between amino acids with a sulfur on the side chain. So like cysteine, it has sulfur in the side chain. Okay. Yeah. So here we have hydrophobic interactions, ionic bonding, we have disulfide bridge, we have hydrogen bonding for polar R groups. This is for nonpolar R groups. This one is for polar R groups. We also have, only for some proteins, something called quaternary structure, which is going to be the result of the union of two or more polypeptide chains. An example is going to be collagen or hemoglobin. In the case of hemoglobin, it's made of 4 of those chains that have been bonded together. Okay. So yeah. Now, sometimes a problem in the primary structure can destroy the entire protein. For example, hemoglobin. Its importance is to transport oxygen. So if you mess up with that function, your body cells will then starve for oxygen. So in this case, for example, the primary structure was affected and the 6th amino acid changed from glue to val. In which structure? In the primary one. What was the thing that determined the primary structure? The DNA, what? Sequence. So if you change that sequence by a mutation, it could possibly affect the primary structure, which means you will change glue to val. Now, how is that important? Glue is a polar amino acid, while val is a nonpolar amino acid. The problem is glue is found on the outside of the hemoglobin. What's also on the outside? Water. So glue being there is okay, but val being there is not okay. That's why now it's going to form this corner point, or it's like a hole, to avoid contact with the water. And now it's going to be forming this sharp edge, which means it will now get stuck inside the capillaries, and the person is going to be suffering from lack of oxygen in their body. That's actually called sickle cell disease because of the shape of the hemoglobin of the red blood cell. It will now become a sickle shape, like a moon. So now it's going to get stuck. So because of one amino acid change, the whole thing changed. And which structure? The primary one, because of a mutation. A mutation in the DNA. Okay. So yeah. Now, how about the protein structure? By the way, if we mention three-dimensional shape, what structure are we discussing? Primary, secondary, or tertiary? DR? You were participating a lot last time, so this time, what? What determines the three-dimensional shape of a protein?
Does its unique shape?
Yeah. Which structure? We got three. Pick from 1 to 3.
The dye one.
It's three: primary, secondary, tertiary.
Oh,
First letter is T. Hint.
tertiary.
Tertiary, yes.
Yeah.
Okay. It's okay. He's stressed about the SAT. Don't worry about it. Get the results soon. Get the 1,250 you're aiming for. Inshallah.
You didn't get it yet?
2 weeks.
All right. So, yeah. See, folding means tertiary structure. Now, if you change the pH, if you change the temperature, if you change the environment, whether it's polar or non-polar, all of that can make your protein unravel, which means it can lose its shape. And once it does, it will lose also its function. So yeah, so the protein loses shape. That's called denaturation. That means it's not going to work anymore. It's going to become biologically inactive. Okay, so to do this, you could increase the temperature beyond a certain point. Maybe you could change the pH. So if the protein likes to work in the basic pH, now put it in the acidic one, it's not going to work anymore. Or maybe transfer it from an aqueous environment, which it likes, which means polar, to a non-polar environment. So if you have a polar protein that likes a polar environment, and now you place it in a non-polar one, it's going to also denature or essentially lose its shape.